Welcome to Unit 2.7 of our Molecular Biology unit. In this video, we'll be looking at the first part of 2.7, which is featuring DNA replication. Okay, so before we actually look at what DNA replication is and how it occurs, you need to be confident at understanding the structure of DNA. And that's what we did in our last unit on where we looked at the differences between DNA and RNA. Okay, so we've seen this comparison between our DNA and RNA, knowing that in the DNA we have the four bases: cytosine, guanine, adenine, and thymine. And knowing that DNA is that double helix. So when we're talking about DNA replication, we need to look at how we can replicate a piece of DNA. So how on a cellular level is a piece of DNA replicated? Because when a cell divides, that DNA needs copies in both daughter cells. So how does that occur? Now, in 2.7, we look at DNA replication, which is how one piece of DNA copies into two identical pieces of DNA. And then in the second video that you'll have on 2.7, we look at transcription and translation, which is where DNA is copied to RNA, and then the RNA is used for protein synthesis. So in this video, we're looking at DNA replication. And that's the same slide. Okay, so DNA replication. This needs to occur so that the daughter cells are genetically identical to the parent cells. DNA replication copies DNA precisely so that the new molecules produced have the exact same sequence of bases as the original stand, and this occurs during the S phase of the cell, the S phase of the interphase in the cell cycle. Okay, so DNA replication, we've got a few steps. So first of all, we have the enzyme helicase that moves along the DNA double helix and literally unzips it. So breaks the hydrogen bond between the complementary bases, between those base pairs, and it results in two single strands then. So the separate sides of the ladder as such. So in this image, you can see helicase there. It's the first thing that's happening. It's going along and unzipping the DNA. Step 2. The single strands are then exposed to unpaired nucleotides, okay, and they act as a template for building a new DNA molecule. So free nucleotides that are within the cell then move into place, so they pair with their partner pair, okay, so that base pairing. So A pairing with T, so adenine pairing with your thymine, cytosine pairing with guanine. Okay, so you can see here that the pairing is occurring. Okay, the pairing's occurring. Okay, so DNA polymerase guides the linking of the nucleotides. Okay, so if we go back and have a look at that picture. So we've got several different polymerases, okay, but they are linking the base pairs. So these free nucleotides to their exposed partner base as such. Base pairing between bases on opposite strands occurs with the formation of hydrogen bonds. So those free nucleotides pair with the base, and those bases bond together with the hydrogen bonds that we've already spoken about. Then condensation reactions occur, creating sugar phosphate bonds between the nucleotides to make new nucleotide strands. So remembering that our polymer of nucleotides is that sugar phosphate bond between the sugar molecule on one nucleotide and the phosphate on the next one. So this then makes our new polynucleotide strand. The two new DNA molecules are now rewound to form two new double helixes. Okay, so the free nucleotides are bonded with their exposed nucleotides, okay, depending on the base pairing, and then the nucleotide, the sugar phosphate bond occurs between these free nucleotides. Okay, so we've got this DNA polymerase that creates a complementary strands. Okay, so this is our DNA polymerase, and it's linking those bases to their parent strand. So it's free nucleotides. So you've got the helicase unzipping the DNA, in come the free nucleotides, and the DNA polymerase then links all those free nucleotides together. Okay, so just a few other things. The base sequence on the template strand determines the base sequence of the daughter strands. We call this semi-conservative replication because you see the two blue strands are the parent strands, and there's a blue strand on each daughter strand. Okay, so it's semi-conservative because it keeps one strand of DNA in the new strands that are formed, in the new double helix formed. Okay, so one of the things we need to know about when we're looking at this area is PCR, so the polymerase chain reaction. Okay, now PCR is a way of producing large quantities of a specific target sequence of DNA. It's useful when only a small amount of DNA is available for testing. So, for example, crime scenes, blood samples, semen, tissue, hair, etc. PCR occurs in a thermal cycler and involves a 3-step procedure which is repeated. So to start off with, we've got denaturation. Okay, now this is the DNA sample is heated to separate the two strands. So remembering when you heat molecules, those hydrogen bonds tend to break. So you heat it up and you break the hydrogen bonds between the strands. We then have annealing, where the DNA primers attach to opposite ends of the target sequence. And then we have elongation. Now this is a heat-tolerant DNA polymerase attack which copies the strands. So we've got two different representations here. We've got the step 1, that denaturation, so it's been heated and it's separated the chains out, so the separation of the chains. The annealing, and you'll see the temperature difference here. Okay, so we've got a forward primer and a reverse primer working. Okay, so we're copying the, we're putting the free nucleotides in now, we're forming the new strand, and then the extension or the elongation is where the synthesized polymerase comes along and causes condensation reactions to occur between those nucleotides, forming the new strands. Okay, so the reason that we do this, as you can see, we've got this initial DNA, and then we can replicate it very, very quickly using the synthesized process. Okay, that's it for the first video. You now need to watch the second video on transcription and translation.